Hi, good afternoon, everyone. And I'm Zhang. I'm a master's student from Auburn University. And the topic today is about physiological responses of peanut varieties to mid-season drought stress. As we all know, in the United States, peanut mainly used for food like peanut butter and snacks. It also can be used for livestock feed, soap, shampoo, and fuel. And in 2020, the United States peanut production was 6.6 billion pounds. And 80% of the peanut are runner type, and they mainly planted in the Southeast area. And Alabama produce around 10% of the domestic peanut productions every year. However, due to the unpredictable weather and the unevenly rainfall event, so the drought stress is very common in Alabama. And a previous study mentioned yield reduced around 56 to 85% due to the mid-season drought through the pot filling stage in this country. And in Alabama, the mid-season drought can have a big effect on the peanut yield, and 90% of peanut field in Alabama are not irrigated. And drought can cause many adverse physiological effects, such as the reduced turgor pressure in leaf, so their leaf expansion inhibited. Therefore, the plant aboveground biomass reduced. And also drought can cause abscisic acid accumulation, which trigger the stomatal closure. Therefore, the photosynthesis will reduced, then the plant have lower yield in drought condition. Well, plants can respond differently under drought in yield. As we can see from the pictures on the right, some drought-sensitive varieties, they produce very little yield under drought condition, while some other drought-tolerant varieties, they can have a high yield in the same environment. So to determine physiological mechanism contributed to the high yield under drought will be very helpful for breeding for the peanut drought-tolerant varieties. So some drought-tolerant strategies is about the water use efficiency. Water use efficiency is the ratio of biomass produced to the rate of transpiration. Previous study has mentioned two contrary drought-tolerant strategies to maintain high yield under drought. And one was called high water use efficiency, also saying water saver, because some drought-tolerant plants, when they detect the drought stress, they will close the stomatal to preserve the water by reducing the transpiration, but the stomatal was not completely closed. So the plant can still maintain a relatively high photosynthesis, and then in the end, plants can still get a high yield in the end of the season. And to test this mechanism, we measured photosynthesis and stomatal conductance in the field, but the results are very variable at different times that we measured in the field. So we use carbon isotope discrimination to estimate the integrated water use efficiency in plant during the whole grain season. The CID is the ratio of is the difference of the ratio of carbon 13 by carbon 12 in the leaf and in the air. So the water saver plants usually show a low CID value, and the other drought tolerant strategy is low water use efficiency. Means they are high water use, so they called water spenders. That's because some drought tolerant plants can develop deeper or denser roots in the soil in the drought condition, so they can maintain the high transpiration. So the opening of stomatal can allow plants to do more photosynthesis. Therefore, plants can maintain a high yield in the drought condition. And the water spender plants usually showed a high CID value. And in this experiment, our objective was to distinguish different drought-tolerant mechanism in high-yielding peanut under the drought. To do this, we selected 36 cultivars to planted in 2019. they were classified as drought tolerant cultivars, moderate drought tolerant cultivars, and drought sensitive cultivars based on Dr. Chen's previous study. And in 2020, we picked, based on the yield performance in 2019, we picked 18 cultivars to plant it in 2020. And this experiment was conducted in the rainout shelters located in the National Peanut Lab in Dawson, Georgia. The rainout shelters incorporated the rainfall sensor, which can help us to control the water stress very well. And to simulate the drought, mid-season drought, we started the drought stress from 70 days after planting to 105 days after planting when the plants were in the pot filling stage. And we set the well water treatment in one shelter as a control. And we used the Lycor 6400 machine to took the photosynthesis and stomatal conductance measurements. And we measured 4 times in 2019 and 6 times in 2020. And we also collected the CID data and the yield data at the final harvest. So for the yield result from 2019, and we find there was a significant difference between different cultivars. And in this graph, we can see in this graph, the red bar means they were drought tolerant cultivars, the yellow means drought moderate tolerant cultivars, and the blue ones means they were drought sensitive. So from this graph, we can see the drought tolerant cultivars, they showed higher yield under drought condition than other varieties. And also you can notice the PI50 and PI49, they showed very high yield under drought condition. And also for the photosynthesis result, and we find there was an interaction between days after drought and genotypes. And in this graph, we mark the drought tolerant cultivars by the solid line, and the dashed line means they're moderate drought tolerant, and the dot line means they are drought sensitive. And we can see the drought tolerant cultivars, they showed higher photosynthesis than other cultivars. And also you can see the PI50 and PI49, they showed high photosynthesis in the drought condition. And also 2 other high yield cultivars, T508 and Li8, they showed a relatively high photosynthesis compared to the drought sensitive cultivars. And we also analyzed the stomatal conductance result data, and the result showed there is no interaction between day after drought and the genotypes. So this graph in the below showed the overall stomatal conductance for each cultivars under drought condition, and also the mean stomatal conductance for all the cultivars in well water condition. So in this graph, we can see the PI50 and PI40 line. They are the high yield cultivars. They showed high stomatal conductance, and other 2 high yield cultivars, T508 and Li8, they showed low stomatal conductance. So combine for the result of photosynthesis and stomatal conductance, we can see the PI50 and PI49, they showed high photosynthesis and high stomatal conductance. They may have a high transpiration, might means they are the low water use efficiency, might be the water spender plants. And the T508 and Li8, they showed relatively high photosynthesis, but low stomatal conductance, which probably means they are the high water use efficiency plant under drought condition. And we can confirm that in the CID value results showing that in these slides. And the seed CID value showed the PI50 and PI49, they have high CID value, means those 2 high yield cultivars, they are low water use efficiency. So they were the high water use, they are the water spender plants. And 2 other high yield cultivars, T508 and Li8, they showed low CID value, means they are the high water use efficiency plant. under drought condition. And also we did the correlation for the CID in seeds and the yield for both years. And there was a positive correlation between them showing in both years. And we can see the PI50 and PI49, they showed high CID value under drought. So they were characterized as the water spender plants. And other two high yield cultivars, Qiong and line 8, they showed lower CID value. So they were characterized as the water saver plants. And the correlation for yield and the mean stomatal conductance over the whole season for each cultivar showed the very high and very positive correlation between them. And this correlation graph explains those two contrary drought tolerant mechanisms under drought. We can see the PI50 and PI49, they have high stomatal conductance under drought. So which means they have high transpiration. So they are the water spender plants. And the other two high yield cultivars, line 8 and Qiong, they showed low stomatal conductance under drought, which means they are the high water use efficiency plants. So they were also called the water saver plants. And there are some conclusions from this study. First, the PI50 and PI49, they have the high yield under drought, showing high photosynthesis and high stomatal conductance. They were considered as water spender plants. And the reason might because they have more access to water due to the deeper or denser roots. And we're gonna study that in the future. And also we find some other high yielding cultivars have high water use efficiency in drought, showing relatively high photosynthesis, but very low stomatal conductance, such as Qiong and line 8. And also we find the stomatal conductance was highly positive correlated with the pot yield under drought condition. And thank you very much. And I'd like to take some questions if you have.
Very nice presentation. And we have a question here from Dr. Cristiani Pilon. Were there any, please leave your screen up there in case we need.
Yeah, I'm sorry.
Question is, were there any innate differences in stomatal conductances or photosynthesis rate among the genotypes under well-watered conditions?
There is no significant difference in well-watered condition because they are all irrigated very well. So there's no difference for sensitive cultivars and tolerant cultivars.
Is that something that you measured, or is that something that's just assumed to be?
We measured, that's why we use the mean value for all the cultivars, because there is no significant difference, and also for the stomatal conductance and the photosynthesis. The dark blue line is well water condition.
Okay, excellent. Yeah. The next question for you, I guess that's no, there are no more questions. Let me check just in the chat. Okay, any further questions for our speaker? With that, I'll thank you for your presentation. Excellent presentation. Thank you for participating in our session this year. Okay.
Thank you.